in the last lecture we had discussed Michaelis-Menten kinetics as applicable to enzyme catalyzed reactions and also the proposed modifications by Briggs and Haldane, whereby he confirmed the formation of enzyme substrate complex and the steady state hypothesis with respect to the formation of ES complex, and that the enzyme substrate complex is in a steady state soon after the reaction starts. After a very brief induction period, the concentration of enzyme substrate complex comes to a steady state. That was the major hypothesis, and based on this hypothesis we had also arrived at a rate expression V naught is equal to K2 E naught S upon Km plus S. Now, I am just writing here only the capital letters for the concentration terms without putting the parenthesis, and here your Km was equal to K minus 1 plus K2 upon K1, whereas Vm was a term which was defined as K2 E naught, and these two parameters, that is Michaelis-Menten constant and the maximum reaction velocity, can be used to characterize an enzyme. I also pointed out that Vm is not a constant, is a function of enzyme concentration, and we must take a corresponding kinetic parameter as K2, which is often called as turnover number. Now, one of the most important features of this kinetics, which I would like to reiterate, are the assumptions that are involved in arriving at these rate expressions, and I would like you to clearly consider them while applying this kinetics to any experimental situation. As we also mentioned in the beginning, that any kinetic analysis will involve the match between the hypothesis, the rate expression and the experimental data, and therefore, whenever we look at any experimental data, we must ensure that under certain set of conditions the assumptions that are built in the Michaelis-Menten kinetics are true and they are met. Now, just to reiterate the assumptions, the major assumptions which were made here are formation of ES complex. Now, basically we have assumed that the enzyme has a single substrate binding site, and on this the substrate molecule binds and a ES complex is formed. This also limits to a single substrate reaction. The kinetics is basically developed on single substrate enzyme catalyzed reactions. It is not really directly it does not directly interpret the multi substrate reactions which are so common in the case of biochemical systems. The third assumption is it talks of initial reaction rate, and when I say initial reaction rate, the emphasis is that the product concentration is almost 0. That means we are talking at the rate when there is no product formation. So, inhibition by the product or the effect of product concentration on the reaction rate has been negligible. And the fourthly, we have also made assumption that the substrate concentration is much larger than enzyme concentration, which is in most cases a very true assumption, and the enzyme concentration being the catalyst is very small compared to the reactant, as is also is the case in most of the catalytic reactions. And the implication of this assumption is the fact that when we monitor our reaction rates, we can safely assume that your S0 is approximately equal to S. That means the substrate concentration at any given time, because not much of product is formed, and therefore such an assumption can be held true. Then, as I mentioned, that the Briggs and Haldane's hypothesis was regarding the steady state with reference to ES complex. Now, this is a very loaded statement in the sense, particularly a deviation from the original Michaelis-Menten hypothesis, which states of the equilibrium of ES. Now, here you must appreciate that when we talk of equilibrium of enzyme substrate complex with enzyme and substrate molecules, it means essentially that the breakdown of enzyme substrate complex into product is a very slow process, and it does not disturb the equilibrium process of the enzyme substrate binding. That is what was the original concept of Michaelis-Menten. But when this was disputed by Briggs and Haldane, particularly observing certain reactions which were known to be very fast, relatively fast, then you cannot say that the breakdown of ES into product is a very slow step, and obviously a reasonable rate of breakdown will disturb the equilibrium, and therefore, what he mentioned that the rate of breakdown of ES and rate of formation of ES are probably at the same rate, and the ES remains under steady state. So, the rate of change of concentration of ES is 0 and the steady state assumption, and that is what probably a very major understanding which we must appreciate, and of course, finally and a very simplistic assumption is that we always monitor or study or consider this kinetics in terms of constant pH, temperature, ionic strength, and all other environmental parameters we try to keep constant, maybe sometimes water activity, and all those parameters are maintained constant. So, I think in the light of our earlier sort of understanding that any kinetic analysis should be made with keeping in view the mechanistic hypothesis, the rate expression, and the experimental data, we must ensure that. Now, for understanding or for probably analyzing the experimental data for any given enzyme catalyzed reactions, we can simply use Michaelis-Menten hypothesis directly, but the use of direct Michaelis-Menten hypothesis or Michaelis-Menten equation has a limitation, because it represents a rectangular hyperbola, where the maximum reaction velocity is a asymptotic situation and you can never experimentally reach that, and therefore, it often creates problem in determining the various reaction parameters, particularly the Vm and the Km values. So, therefore, the simplistic way, as we were also talking yesterday, was to linearize the Michaelis-Menten equation, and one of the simplest way to linearize was double reciprocal plot. That means you take reciprocals on both the sides of the expression, that is your 1 upon V naught is equal to 1 upon Vm plus Km over Vm into 1 upon S. Now, such an equation, which is a double reciprocal on both the sides of the Michaelis-Menten equation, represents a straight line, and it can be, the experimental data can be easily analyzed based on such a 1 upon S profile versus 1 upon V profile, and the expression will represent a straight line with a slope of Km over Vm, and intercept on the x axis as 1 upon Vm, intercept on y axis as 1 upon Vm, and intercept on x axis as minus 1 upon Km. Now, this was one of the earliest analysis for the experimental data in the light of Michaelis-Menten hypothesis. Now, it was noted to have very serious limitations, and the serious limitations are that if you look at the data, you take any set of data for enzyme catalyzed reactions, that means data on the substrate concentration versus reaction velocity, you will notice that the experimental data gave very high or very low weightage to the measurements at low substrate concentrations. That means most of the data points will be rather concentrated near the origin, most of the data points, and whereas here they will be at a very, the weightage at a high substrate weightage at a low substrate concentration will be very low. There will be under weightage at the low substrate concentrations, and at high substrate concentration, that means near the origin, the data point will be heavily concentrated. And you can just see, I mean just numerically, just for example, if you consider substrate concentration in millimolar quantity and the reaction velocity in some arbitrary units, you see at 5 the 1 upon S will be 1 upon millimolar will be 0.2, 10, 0.1, 15, 0.66, 20, 0.05, 30, 0.033, 40, you come to 0.025 and so on. So, you see that when you increase your substrate concentration, the weightage to 1 upon substrate concentration is so low that ultimately you end up in scattering a very, I mean cluster of data near the origin, and therefore getting the realistic value of Km and Vm often tends to be difficult even after getting a statistical, making a statistical analysis. And therefore, a number of other linear plots were thought of, and the other linear plots are Haynes plot, which is essentially nothing else but the modification of the original equation. That means, if you multiply the Lineweaver-Burk equation simply by substrate concentration on both the sides, you arrive at S upon V naught equal to Km upon Vm plus S upon Vm. Again, this also represents a straight line, and if you see a graphical representation of such a plot of substrate concentration versus S upon Vi or V naught, you will again end up in a straight line equation with a slope of 1 upon Vm and an intercept of Km over Vm. And of course, the intercept on the x axis as minus Km. Now, again here you have a little better situation in the sense that the data are usually not so clustered, they are scattered because you have substrate concentration on both the axes, and therefore you get a much better fit. And a similar profile, an alternative profile is Addy-Hofstee plot. Another version of the linear form of Michaelis-Menten equation, where you can arrive at V naught equal to Vm minus Km upon S into V naught. So, if you plot such a equation, and that you can obtain by multiplying the Lineweaver-Burk equation by V naught into Vm on both the sides, and then you get a straight line with a negative slope, where slope is minus Km. Of course, the axes here are V naught upon S versus V naught, and you get the slope of minus Km, and the intercept on the y axis is Vm itself. This also has the advantage of almost well scattered or data points, and that can be easily used. Now, in all the three sort of linear plots which I discussed, that is Lineweaver-Burk plot, Addy-Hofstee plot as well as the Haynes plot, again there is one limitation which has been noticed. We have no means to look at the adequacy or the effectiveness of our analytical procedure which we have used, because after all the accuracy of our determinations or experimental analysis of experimental data will largely depend upon the accuracy of our experimental methods, and whatever data point we get, we can always make a statistical fit into form of a straight line and get the values. We do not get any insight into the correctness of the data. In fact, to take care of that, another important plot which was proposed not very late, it was in 1974 that Bowden plot, or rather Cornish-Bowden plot. It was developed by two gentlemen. Cornish-Bowden plot was developed in 1974, as late as 1974, and this had a characteristic feature in the sense that if you write the Michaelis-Menten equation assuming the substrate concentration and the initial reaction velocity as constants and Km and Vm as variables, although it may look little I mean sort of unusual, unrealistic, but just for the sake of a mathematical analysis, if we write that, we can write for example 1 upon V naught as equal to Km plus S upon Vm plus Vm into S. Now if you just take a reciprocal, and therefore Vm upon V naught will be equal to Km plus S upon S, and therefore your Vm will be equal to V naught into 1 plus Km upon S. Now this also again represents, you notice that if we have just written the equation in the form of Vm and Km as the variables. Now this also represents a straight line. Essentially if you look at Vm and Ks will also represent in a straight line, and with a slope of 1 upon S and intercept of V naught. Now as I mentioned that it may look little unorthodox, and because Km and Vm we know are constants, I mean at a constant enzyme concentration Vm is also constant, but we can consider that if we consider a single data point, that means at any given point of S1 we consider V1, another set of conditions S2 we consider V2, for each of these two data points the straight line relationship between Km and Vm must be valid, at least from mathematical point of view. So what we do is in this case that for each set of data point between S and V, we develop a straight line. That means we simulate the value of V, I mean we just assume a hypothetical value of Km, corresponding Vm is calculated, and you make a table, and we tend to sort of draw a straight line with reference to, let us say, S1 V1. Similar exercise we do for the second data point. We also develop another expression, another straight line, and this gives you, let us say, for S2 V2. Third point we also develop S3 V3. So for a number of data points, for each data point we can develop a straight line. Now essentially the enzyme system, the reaction will have only one set of Km and Vm, and that will be the one which is obtained by intersection of these straight line relations, and that gives you a perfect check on your experimental data. Very often when you will probably sometime notice in the experimental data, you may not get a perfect point of intersection. The point may slightly, let us say, go something like this. Then you are sure in one thing that your Km and Vm values are somewhere in this range. You know the range of Km and Vm value, not the absolute value, and you also know the limitations of your experimental method that you are following. There is somewhere something flaw which has given, because theoretically if your all data points or measurements of substrate concentration and reaction velocity are perfect, they must intersect at each other. I am assuming that Michaelis-Menten kinetics is valid. So basically Carney-Bowden plot gives you an opportunity to not only get the range of Km and Vm values, but also makes a check on the validity or accuracy of your experimental data, and which is probably one of the very basic features in the case of enzyme kinetics, that your experimental data must be pretty accurate to be able to analyze the kinetics of enzyme catalyzed reaction. So I think using either of these plots, and personally I recommend usually Carney-Bowden plot for any analysis, we must be able to analyze or get the correct kinetic features or characteristic of any enzyme catalyzed reaction. Of course broadly in terms of the value of K2, that is what we call as turnover number, and Vm and Km value. Now there is another very interesting feature as far as the Michaelis-Menten equation is concerned, and some of the very interesting features you will notice that when we mention our assumptions of Michaelis-Menten equation, you will notice that these assumptions will largely not fit into most of the actual enzyme catalyzed reactions. This is so happen, but still the Michaelis-Menten equation is still considered to be the most universal sort of image or most sort of universal model for analyzing enzyme catalyzed reactions. Now obviously there is somewhere some compromise, and essentially if you look at each of the points where we have paradoxes in terms of the actual enzyme catalyzed reactions and those which can be done by Michaelis-Menten kinetics. The first one, that the Michaelis-Menten kinetics deals with the single substrate enzyme catalyzed reaction. Now I think with your background in biochemical metabolism, you will appreciate that most of the enzyme catalyzed reactions in practice in the living cells are not single substrate reactions, they are multi substrate, at least two substrate reactions, and if not any other directly converting substrate, water is a molecule which participate in the reaction. Now in such a cases the applicability of Michaelis-Menten equation is still valid under conditions that one of the substrate is held at a constant concentration which is in excess constant concentration. See, one substrate which is limiting, I think we here would like to define that the Michaelis-Menten kinetics should be applied to a limiting substrate, and the other substrate must be in excess, and therefore its concentration can be considered to be constant. That means it does not, even after it is consumed partly, the concentration drop is not very large. And actually if you look into most of the hydrolytic reactions, the water which is almost about more than 50 molar concentration in the reaction mixture, even after participation of water in the hydrolytic reaction, the concentration does not drop to anywhere significant level, and therefore Michaelis-Menten kinetics still holds valid and gives you on analysis practical results. The second limitation and of course the significance of Michaelis-Menten equation comes that it assumes one substrate binding site per enzyme molecule. When we wrote our mechanistic expression enzyme substrate binding, we are only taken one enzyme, one substrate molecule binds to one enzyme molecule, thereby assuming that the enzyme possesses only one substrate binding site, which is also not true in many cases. Most of the enzymes, and particularly the ones which have quaternary structure for their function, they will have more than one binding site on the substrate on the enzyme molecule. But such a fallacy can be taken care in the light of Michaelis-Menten kinetics if the binding sites, even if they are more than one, if they are not interactive, that means they are independent of each other and do not interfere in binding of each other, then the Michaelis-Menten kinetics will be valid. The only difference will be that you must take into account of multiple binding sites in terms of the concentration of enzyme, that in concentration of enzyme will reflect the concentration of active sites. And the third is, we have talked about formation of single intermediate complex. When we say your enzyme substrate complex is formed, we are talking only that one enzyme substrate complex is formed. Now in many cases, even which are now practically understood very clearly, this is not true. Very often we end up with kind of interactions as E plus S going to ES, going to ET, going to EP, E plus P. That means or alternative sequences, there are a variety of intermediate complexes that are formed, not only one complex is formed, and this is very true very often in the case of reversible reactions. And now in such cases, the Michaelis-Menten kinetics will also be valid provided the interconvergence of the intermediate complexes are the rate limiting steps. Now if you just simply consider this as an irreversible process, and this is K1, K minus 1, K2, K3, if this K2 is the rate limiting step, you must identify only the rate limiting step and take Michaelis-Menten kinetics in the light of the put the rate constant K2 that appears in the Michaelis-Menten kinetics as one, which is the rate limiting step, and then Michaelis-Menten kinetics will apply to it. And then you see many of the enzyme catalyzed reactions, they involve, today we know that they involve more than one intermediate complexes, not just one enzyme substrate complex. Now the Michaelis-Menten kinetics can still apply to those reactions, provided the interconversion of the complexes is assumed to be the rate limiting step, or in other words, in place of your turnover number K2 that appears in the case of Michaelis-Menten kinetics, you put the rate limiting rate constant in the sequence of interactions in the enzyme substrate or in the enzyme kinetics, and that will still be valid. Related to the same issue of multiple intermediate complexes is another is turnover number may involve several individual rate constant. We have often defined our the turnover number Vm is equal to K2 into E naught, and this K2 essentially is being defined as the turnover number. Now, in most of the enzyme catalyzed reactions which involve multiple intermediates, a single rate constant may not be able to define the turnover number. Turnover number essentially may work out to be a lump parameter consisting of more than one rate constant. Just for example, you consider a very practical case which has been studied extensively, like chymotrypsin catalyzed hydrolysis of esters and amides, and if you look at the reaction sequence which it follows today as understood, ES going to EAC going to E plus of course P. Now, if you look at your K1, K minus 1, K2, another product is formed, this is P2, this is P1, there are two products formed on hydrolysis, both one acid as well as an alcohol, and this is your K3. Now, if you just analyze this by steady state hypothesis and take steady hypothesis with respect to the steady state with respect to the EAC, which is the acylated enzyme complex, you get the reaction velocity as E naught into S into K2 K3 upon K2 plus K3. In fact, just for when we will proceed further, we will develop a system by which we can write the expression for more complicated kinetics, because when we write for single substrate complex, the single enzyme substrate complex, the developing the steady state rate expression is very simple. As you reach to more and more complexity in terms of more enzyme substrate complexes, the mathematical analysis will become more and more complex, and there are routes by which one can write straight away by observation the rate expression, and we will discuss that in the subsequent classes. And this upon K minus 1 upon K1 upon K2 plus K3 plus S. Now, here in such a case, your K catalytic, in analogy to the original Michaelis-Menten hypothesis, will be equivalent to K2 K3 upon K2 plus K3, not a simple K2. The turnover number essentially will depend upon both the parameters K2 as well as K3, and that is another fallacy which has been which is built in the Michaelis-Menten kinetic. It does not take care of that. So, basically the Michaelis-Menten kinetics need to be extended to develop a rate expression which takes into account steady state with respect to one of the intermediates and analyze it in analogy to develop a lumped parameter for defining the turnover number. Now, the turnover number for most of the enzyme catalyzed reaction varies in the order from 10 to 10 to the power 4. per second. That is the order which it goes. I mean, it is fairly large range. So, basically that also dictates that there are certain reactions which are slow, there are certain reactions which are very fast. A reaction with a turnover number of 10 to the power 4 per second, that means so many number of molecules of substrate converted to product per mole of the enzyme, is a very large turnover as against a turnover number of 10. Now, the next and a very significant advantage of Michaelis-Menten kinetics, and particularly significance of the Km Michaelis-Menten constant, is we very often get an insight into the functioning of enzymes in the living cell. Take the simplest example, glucose 1-phosphate. I mean, glucose is the starting point when we go into the metabolism, and it's the first step is phosphorylation of glucose, and you get a series of products, glucose 1-phosphate, glucose 6-phosphate, or fructose 1-phosphate, and so on. And now these molecules will essentially can go further either into the glycogen synthesis as a storage molecule, or further to fructose biphosphate. Now, essentially the route which these molecules will follow will depend on the relative Km value of the two enzymes involved. The enzymes, the Km value for the fructose biphosphate forming enzymes is much smaller, at least 1,000-fold smaller than the glycogen synthesizing route, and therefore, as long as the concentration is of the these molecules phosphorylated glucose is not very high, it will always tend to go into the fructose biphosphate route, and therefore and release the energy. Only when the concentration of this phosphorylated glucose becomes quite high, a part of it, depending upon the Km, the ratio of the Km value, will go to glycogen synthesis. So, that is one simple example, but at many points where the substrate has more than one route to follow in the biochemical metabolism, the its preference or the likely physiological route can be decided on the basis of Km value, and that has been possible basically based on the understanding of Michaelis-Menten kinetics. Another important significance of the Michaelis-Menten kinetics is its application to assay of the enzymes. I think one of the analytical advantage, you see, very often we tend to we are required to estimate the activity of an enzyme sample, and this enzyme sample activity must depend upon the effect of enzyme concentration on the reaction velocity, and essentially we use such a parameter, particularly when we plot the concentration of enzyme on the reaction velocity. We get a profile, again initially it is a straight line which is linear, and then it tapers off towards the end when the enzyme concentration becomes high. Now, normally we assay the enzyme concentration in our linear range. There are two parameters, there are two constraints which have to be strictly obeyed when we talk of assay of the enzyme, rather three constraints. The first constraint is the detection of the product. I mean, obviously detection, you must have a method for detection of the product formed of the reaction. Now, the limitation on the detection of the product, what is the minimum quantity of product you can detect, will dictate the time of the reaction. That means, how much time reaction to carry out will be considered on the basis of the detection method for the product, so that you can accurately determine the product. The second limitation is that you must follow the linear range of enzyme reaction velocity file, the linear range. The third parameter is, one must ensure that what should be the substrate concentration for assay of the enzyme, and for all analytical purposes, for all assay purposes, the substrate concentration that is taken is much larger than your enzyme concentration, and for all practical purposes it is taken the reaction velocity what we measure is essentially V naught must approach to V max. That means the substrate concentration is very large. Now obviously as I mentioned that we can never reach effectively to Vm, we can only approach to Vm, and for all practical purposes the substrate concentration that is chosen is roughly of the order of about greater or equal to 100 km values. So the magnitude of Km, magnitude of Michaelis-Menten constant also gives you a parameter for assay of the enzymes, so that you can choose your substrate concentration for carrying out the experiments, and that is another major sort of significance of the Michaelis-Menten kinetics. Another important feature of Michaelis-Menten kinetics is to define a specificity constant. Now if suppose we have if an enzyme is active on more than one substrate, now how to define its specificity or compare the specificity of the enzyme with respect to different substrate, and the kinetic parameters available from Michaelis-Menten kinetics can give you some insight into this. Now if you consider, as I mentioned earlier, if you consider at a very low substrate concentration, when substrate concentration is much smaller than Km, then your initial reaction velocity will be equal to K2 E naught into S upon Km. A first order kinetics primarily, and basically with this as a pseudo first order rate constant Kf, and therefore one can assign or it can consider K2 into E naught, or E naught is again, if you consider variable, then K2 upon Km is essentially a constant which is considered as specificity constant E naught into S. I mean very often sometimes some people call it a pseudo second order rate constant, considering enzyme also as one of the reactants, which is not really second order rate constant, and K2 upon Km is considered as a specificity constant which determines the relative specificity of an enzyme to different substrates, and one can determine these values from the kinetic analysis, and one can get the picture of specificity. Now under this first order regime, or under the conditions of S0 very small than Km, there is another feature which we must look at. At this feature, the E naught is approximately equal to E. The initial enzyme concentration, we are assuming that substrate concentration is very low. So if we can also go to a level that not all the enzyme is saturated, all the enzyme is not bound, bulk of the enzyme is still available in the free form, and therefore the specificity constant relates initial reaction velocity to the concentration of free enzyme rather than the total enzyme. So these are some of the practical implications of the significance of Michaelis-Menten kinetics, which applies to most of the kinetic analysis. Now I think one of the most important feature here, what someone you may be experiencing, is that how do we test the validity of steady state assumption. I mean we have been always assuming, okay, Briggs and Haldane gave it, and believe that lot of people have the steady state hypothesis have undergone very severe tests, both analytical as well as experimental, and it has proved those tests, and that is why it has become so much universally valid today. Now how to analyze that, or rather what are the constraints, or what are the experimental constraints within which the steady state hypothesis must be considered valid, and that you can very easily arrive at, that when you write down the sort of rate expression for rate of change of enzyme substrate complex. For example, if you look at your the original equation that we discussed, E plus S going to X going to E plus P, if you consider the original equation that we mentioned earlier, and we wrote the rate of change of concentration of X. I am again not writing the parenthesis, but the letters, capital letters, denote the concentration terms. Now, they can be written as K1, K minus 1, K2, your K1 into E into S minus K minus 1 X minus K2 into X, now which will be equal to your K1 E0 minus X into S minus K minus 1 X minus K2 into X. Now, you can separate the variables X and T in this equation, and ultimately therefore, you can write dx over K1 E naught S minus K1 S plus K minus 1 plus K2 into X equal to integral dt. Now, if you solve, now if you just solve it, you will get a logarithmic term, and the logarithmic term will be, the solution will be ln K1 E naught S minus K1 S plus K minus 1 plus K2 X upon minus K minus 1, minus K1 into X plus K minus 1 plus K2, that is equal to T plus alpha. Let us say alpha is the proportionality, alpha is the constant for integration. Now, this alpha can be easily determined at T is equal to 0, X is equal to 0. At initial time there will be no enzyme substrate complex, and that can give you simply the value of alpha as ln K1 E naught into S upon minus K1 S plus K minus 1 plus K2. So, now you can substitute the value of alpha and thereby get your final solution to the two integrals, and if you take exponential on both the sides and simplify, you will come out with an expression V is equal to V, you can write as K2 into X, and V can be written developed as, if you just do 2 or 3 iterations in between, you will arrive at Vm into S 1 minus exponential K1S plus K minus 1 plus K2 into T upon Km plus S. Now, which if you consider that if this exponential term exponential minus K1S plus K minus 1 plus K2 into T, if it is, if the value of T is very large, let us say if we consider T very small, that means right in the beginning of the reaction the steady state hypothesis is not valid. It has to undergo, the reaction has to progress to an extent that the T gets a magnitude, and then the exponential term will tend to go towards 0. If the T become large, now the requisite size of the T will depend upon the magnitude of K minus 1 S, K1 S plus K minus 1 plus K2. The magnitude of this parameter will essentially dictate what should be the size of the T, or in other words, this T essentially is nothing else but the induction phase. What should be the induction phase after which the reaction measurement should be made, so that steady state hypothesis is valid. Now, for all practical purposes, people have by rapid mixing techniques, people have been able to analyze and determine the value of K minus 1, K1 and K2, a reasonable value, and particularly if you assume that K2 is a turnover number, and we mentioned that the value ranges from 10 to 10 to the power 4, a reasonable magnitude of this will be, let us say, about 1000 per second, and even at this 1000 per second, the exponential, and of course for time is equal to, let us say 5 minutes or 300 seconds, the magnitude of this exponential will be less than 0.01, a very small quantity, and then this rate expression will essentially get turn up into a Michaelis-Menten kinetics, that is your Vm into S upon Km plus S. That means if the magnitude of this exponential is negligible, now this magnitude will depend on two factors, one is turnover number, and as we already know that the turnover number of these reactions is pretty high, orders of magnitudes of a few thousand. Only then it depends on magnitude of time. If you take reasonable time, then the multiplication of turnover number, or rather not really exactly turnover number, but the sum total of K1S plus K minus 1 plus K2, out of which only one factor is turnover number, that magnitude, if it becomes large enough so that the negative of, so that the exponential of its negative is approaching 0, our system will turn up into a Michaelis-Menten expression. And so essentially the constraints for the applicability of the Michaelis-Menten hypothesis, particularly steady state hypothesis, I will say rather to be very implicit, will be valid in case when we take a reasonable time period and also the turnover number is high. And in all the in most of the practical cases where reactions have been studied, the two parameters have been found to be valid, and that is what it gives a validity to the steady state hypothesis for all practical purposes. And also we saw that in many of the critical situations where the assumptions of the assumptions of the enzyme catalyzed reactions were not valid strictly, under certain set of conditions the Michaelis-Menten kinetics could be considered to be applicable. So I think we stop at